All right, so up to this point, we've talked about different aspects of the central dogma. We've talked about DNA replication. We've talked about transcription and transcriptional regulation. And now we'll be talking about the final end of central dogma, where we talk about protein synthesis and translation, and talking about that in the context of ricin and ricin poisoning. Ricin comes from these castor beans that are part of the castor plant. It's indigenous to the Mediterranean and eastern parts of Africa. And the bean itself produces two different things. It can produce castor oil, which in and of itself is not harmful. It's used for lots of different industrial applications, as well as some therapeutic applications. There are some that try and use it to induce pregnancy or induce labor. But the oil itself is oftentimes used and is not harmful. But oftentimes what happens is, after the oil has been extracted, what's left behind is a water-soluble portion that is the ricin protein. That is the highly cytotoxic or cell-killing portion of the castor bean. And so it's not an insignificant portion of the castor bean, 5% by dry weight. And typically what ends up happening is that in response to exposure to ricin, as you can imagine, it's cytotoxic. It leads to vast cell death, and is tied to GI issues, dehydration, and oftentimes can lead to death fairly quickly. What's also important to note is that it doesn't take very much for ricin to be lethal. It requires a very little amount. And oftentimes when you see assassinations in movies where someone is pricked, for example, in Mission Impossible or in some other Russian espionage movies where someone's pricked with either a needle on a ring from a handshake or from the tip of an umbrella, a lot of that is derived from this really famous assassination of a Bulgarian dissident from the Soviet Union, who in London was pricked by a needle on the end of an umbrella, and with just 200 micrograms of ricin was killed. And in addition to that, a more recent example of this was in 2018. President Trump and General Mattis were both sent castor beans. When if the bean is chewed or if the bean is broken open, that's and if it's consumed, that can be lethal unless there's hospitalization. And so the use of ricin as a poison and as a assassination tool has a long history, as well as a more recent one as well. The reason why we're talking about ricin in the context of translation is that ricin is able to permanently disable ribosomes and therefore halt and stop translation. So to now pivot and try and understand translation, what's important to note is that we've talked about the genetic code from the context of DNA and how that can lead to mRNA transcripts. And now what's important to note is that there are specific mRNA sequences that correspond to specific amino acids. And so these sequences on the mRNA, the way we refer to them are usually in triplet codes. And so you can use this as a key or as a tool to try and go through and look at some of the amino acids that we talked about from the very beginning of the semester. In each triplet, we usually refer to as a codon. For each codon, we have four, for each RNA molecule or base pair within the codon, we have 4 possible nucleotides. So we have 4 to the 3rd possible combinations, yielding 64 possible different combinations here. But as you can tell, or as you can perhaps recall, we don't have 64 different amino acids. And so there's an explanation for that we'll get to later on in this lecture. And as I mentioned a moment ago, a group of 3 bases is referred to as a codon. And that's at the mRNA level. So as you can see here, there are there are instances where multiple codons can encode for one amino acid. We refer to that property as the degenerate property of mRNA codons. What that means is, again, a single amino acid can be derived from multiple different codons. However, that's the degenerate side. There's also an unambiguous, or rather more specific side, where typically a specific codon will always yield or lead to a specific amino acid. So while one amino acid can come from, for example, we can look at leucine in the left column, second row. There are 4 different codons. Oh my goodness, sorry. There we go. There are 4 different codons that can, during translation, lead to a leucine. However, it's not that the CUU codon can lead to different amino acids. It will always lead to leucine. That always leading to leucine nature of a specific codon we refer to as ambiguous or specific. And what's also important to note is that this occurs in a non-overlapping manner. So by nature of how our ribosomes are structured during translation, there's a defined reading frame of a single codon at a time. And once that codon is translated, it'll shift over to a new reading frame of a new codon of new 3 RNA nucleotides. Yes. Yes, there are more possible codons. That goes back to the previous slide where we said there are 64 possible codons. Yes, exactly. And we're going to describe why that's the case in a couple slides from now. So again, we define the reading frame is 3 RNA molecules, and so that's defined by how translation is occurring with the ribosomes. And we have 61 codons that typically encode for an amino acid. Excuse me. The remaining 3, we said there are 64 possible combinations. The remaining 3 encode for stop codons. And so you can see above to the top right that there are specific codons that encode for what we call a stop codon that terminate the synthesis of a peptide chain during translation. And what's also important to note is that there's one specific codon that I will ask you to remember. I'm not going to ask you to memorize all 64, but I'll ask you to remember one. That's the start codon that encodes for methionine. And so one way that that's always helped me remember, there's always little tips and tricks you can use. It's a start codon, it's AUG. School always starts. So the start codon, AUG, that's the one codon I will ask you to memorize. Excuse me. So we are going to quickly look at the stages of translation from a high level big picture view, and then we'll go into each of these stages and provide a little bit more detail. So typically during translation, we have our mRNA molecule that's been transcribed and shuttled after it's been processed to form a mature RNA transcript with its 5' cap that's been sliced and has a poly-A tail. It'll be shuttled out to the cytoplasm, and it will engage with the small subunit of a ribosome. And once that happens, the large subunit will come in and you'll have the first transfer RNA, tRNA, that comes in at the start codon to help initiate translation. And so as I mentioned before, AUG is a start codon that encodes for methionine. Our first amino acid during eukaryotic translation will be a methionine. And so what will end up happening is that we have these 3 compartments within the ribosome that you can see depicted in each image. And we're going to talk about what each of those different compartments are for, but they each constitute the reading frame. They can possess one codon at a time. And so what will end up happening is that the first transfer RNA that encodes for methionine will be moved to that middle reading frame. A subsequent new transfer RNA will come in that will sample that site. And once there's appropriate matching that occurs there, the amino acid will then be added on to the existing polypeptide chain. And eventually what will end up happening is that there's going to be not a transfer RNA, but another factor called release factor that recognizes the stop codon, or one of our 3 stop codons, that then terminates translation and mediates the release of the ribosome from the mRNA and the release of the complete polypeptide. So in addition to going into detail on each of those phases, we're also going to try and provide a little bit more detail on each of the characters that are involved with translation. The first is we're going to be looking at, the first is try and focus a little bit more attention towards the transfer RNA. And so the transfer RNA translates the specific codons and provides the amino acid that's encoded for it. And transfer RNA has a bit of a unique structure. It has these 3 hairpin loops. And there is, via an ester bond, it has specific amino acids that are bound to the 3' end of the transfer RNA. The specific site where the transfer RNA interacts with our mRNA transcript is this bottom region here. We refer to that these specific 3 nucleotides. We refer to that as the anticodon, because it's going to be anti-parallel and complementary to the codon on our mRNA transcript. The top portion at the 3' end, we refer to that as the acceptor stem, because that's the site at which amino acid will be added onto, where it is encoding for that specific codon and mediate the addition of that specific amino acid. And that occurs via an ester bond at the 3' end. And so I've drawn up a really rather crude image. I'm a scientist, not an artist, so I apologize for that. But we're trying to come back and refer to this image. And so, so we have RNA that has a 5' end and a 3' end with the amino acid bound to it. And the specific interaction that's going to happen here is going to be happening these 3 nucleotides that constitute the anticodon, and that will be interacting with 3 nucleotides here that constitute our codon. And so they're going to be complementary but anti-parallel. But that the interaction will be a little bit different, because these aren't, we're not unwinding a DNA double helix with an RNA polymerase to mediate this. And so this is what the transfer RNA looks a bit more in a 3-dimensional conformation. And again, we'll have the mRNA with its specific codon that's going to be complementary and anti-parallel to the anticodon at the bottom stem of a transfer RNA. So earlier it was asked why, or rather how, multiple codons can encode for a specific amino acid. And that occurs because of one of the specific nucleotides on that anticodon region on transfer RNA. There's what's important to note is that we do not have 61 different transfer RNAs, but rather what ends up happening is that there's this diversity that's been recognized that we refer to as the wobbling. And so, what that means is that the base pairing in the first position of the anticodon, so going from the 5' end, this end here will interact with the 3rd base pair on this codon. And what ends up happening is that that base pairing is not strict, and there's a lot of flexibility there. And what ends up happening is that multiple codons can be recognized by one transfer RNA as a result of that, because there's one specific, there's flexibility in the base pairing that can occur at the wobble location on the anticodon. But in addition to that, we have another nucleotide called inosine that can actually bind to 3 different nucleotides, U, C, or A. So this will end up, this flexibility leads to the degenerate nature that we see in mRNA. Yes?
So because of the wall in the mRNA, is that why there's multiple codons that can code for each amino acid?
Exactly, yeah. And so, and that's that, and that nature that we refer to as the degenerate nature of the codons. And so the way we derive inosine is that it's only found on transfer RNA. As hopefully as you can appreciate now, we don't see an I nucleotide in DNA or in RNA, but it only exists, or in mRNA, sorry. It only exists in transfer RNA. So for inosine, it's typically only found in transfer RNA, and it emerges from a deamination event on adenine to produce inosine. And so it can base pair with either U or C or A, and that greater flexibility that this nucleotide provides sets us up where if there's an anticodon that's IAU, it can be complementary to and bind with codons that are AUU, AUC, or AUA, because it can bind to either U or C or A. And that's how we get the degenerate nature, where we can get multiple, and all of these codons here encode for isoleucine. And that's how we can get multiple codons that can encode for a different amino acid. That's one way that we can. So I was describing just a moment ago how there are specific enzymes that define what codon or what anticodons correspond to a specific amino acid. And so what we have for the 20 amino acids that we have 20 of this class of enzymes called aminoacyl tRNA transferases. As I've described before, for any enzyme, you can kind of read right to left to get a sense of what exactly it does. So it's a transferase. It transfers or passes something over to tRNA, and it's passing over an aminoacyl or amino acid. So this is the enzyme that mediates adding an amino acid to a specific tRNA based on its codon. And you get one species of this transferase for each amino acid. So we have 20 amino acids. We have 20 species or types of this transferase. And what ends up happening is that this is this requires energy. This requires an ATP molecule. So we have an amino acid binding domain, an ATP binding domain, and a tRNA binding domain. So what ends up happening is that an amino acid that is specific to this transferase will bind to the amino acid binding pocket. You'll get an ATP that is hydrolyzed, and the 2 phosphates are released. And what ends up happening is that the ATP is used to mediate the binding of the amino acid, the ester bond, to the 3' end of the transfer RNA. And so what's important to note is that we talked about in I think the very first lecture about peptide bonds that exist between a polypeptide chain. And the peptide bonds that exist are derived from the energy of this bond here between the amino acid and the 3' end of the transfer RNA. So what ends up happening is that with each amino acid that's added on during translation, it's leveraging the energy from this ester bond here that was derived from this ATP hydrolysis. And so one of the key takeaways that I want you all to remember from this slide is that we have 20 amino acids and we have 20 transferases with a species of each being matched to a specific amino acid. We have an amino acid binding domain, an ATP binding domain, and the transfer RNA. And the transfer RNA, the anticodon, is specifically recognized at this bottom end of the binding pocket and looks for a matching pair, and then we'll add the corresponding amino acid to create what we call is a charged transfer RNA. not charged as in electrical charge, but rather in an active state possessing the amino acid. So whenever you see or read or hear me saying a charged transfer RNA, that just means a transfer RNA with an amino acid. So looking at the mRNA, that is the template. Yes, Leon.
For the transferase, is that happening while the RNA? Like when is the transferase binding to the
When is the amino acid binding to the transfer RNA, or when is the tRNA binding to the RNA? The latter. The transfer RNA is binding to the RNA during both initiation and elongation. So there's a specific transfer RNA for methionine that's brought in during initiation, and then there's going to be subsequent other transfer RNAs brought in during elongation. What's going to happen is that we'll talk about this later on in the lecture today. There's those three compartments of the ribosome. The last compartment, we call that acid, is kicked out, and now that transfer RNA is going to go to is going to be recognized by a transferase, and amino acid will be added on, and now it's charged again and can be recycled during translation. So that's kind of like a quick snapshot of like the life cycle of a transfer RNA. So now to look at the mRNA from the ribosome's point of view, I'm going to be focusing specifically on eukaryotic mRNA, despite there being both eukaryotic and prokaryotic here. So I want you to focus your attention on the bottom portion of the screen when looking to the figure. We have a start codon. We have our stop codon. We also have our 5' cap and our poly A tail, 5' cap and poly A tail. There, if you'll notice, there's a region between the 5' cap and and the start codon. We refer to that region as the 5' untranslated region. That's everything before the start codon, but after the 5' cap. And similarly, on the other end, between the stop codon and between the poly A tail, we have a region that we call the 3' untranslated region, or we can call them the 5' UTR and the 3' UTR. And in between the start codon and stop codon, that's our coding sequence. That's what's going to be that's going to be everything that's used to create the polypeptide chain. Now, there is an understandable question of why do we have a 5' UTR or a 3' UTR? So the 5' UTR, as we understand it so far, is typically associated with translational efficiency. It's typically based on the structure, based on its structure and based on its interactions with some of the machinery during translation. It can help regulate how quickly and how easily translation is occurring. Similarly, the 3' UTR is oftentimes associated with transcriptional regulation and helping with the helping prohibit mRNA degradation or helping with the stabilization of the mRNA transcript. And so these are both still active areas of research of how the 5' UTR and 3' UTR contribute to these processes, but so far that's the big picture of what we understand at the moment. So now we're going to pivot and look at what the ribosome is doing. So the ribosome is consisted of, as from our big picture slide, 2 subunits, a large subunit and a small subunit, but it's also comprised of ribosomal RNA and ribosomal protein. The ribosomal protein in this, as this illustration shows, is largely the magenta in the figure here, whereas the ribosomal RNA is the silver portion. And so what's important to know is that the ribosome is mostly ribosomal RNA. And so there are some ribosomal proteins that help with the contribution of the ribosome during translation. But what's important to note is that this is where ribosomal RNA, as we've talked about in the past, is present. So we have the subunits. The way in which we talk about them is typically defined by, here we have 50S for the large subunit and 30S for the small subunit. In bacteria, for eukaryotes, it's 60S and 40S. This S is called the Svedberg unit. You can probably determine the scientist that defined the unit. The way this is determined is not by molecular weight, but rather pelleting down or the centrifugation of these ribosomal units. And as you can imagine, the larger the subunit is, the more quickly it is centrifuged down. But this is at hundreds of thousands of Gs. And so the first to pellet down are the larger subunits, and then after that, the smaller subunits. And the S is defined by how they pellet down. And so what's important to note is that the 50S and 30S, when they are in when they're in a complex together, when they're spun down, it's not 80S, but rather 70S. Again, this is not defined by molecular weight, but rather how they're spun down. And so with our ribosomal units, we have a few different sites that are important for us to keep in mind. We have the mRNA binding site that exists on the small subunit. And then within the larger subunits, we have the A site, the P site, and the E site. The A site is where the charged RNA comes in and is sampling the codon and trying to assess whether it's the right match. In the P site, we have the growing polypeptide that's present in that site. And then in the E site, that's the empty or the exit site. That's the site where the uncharged or empty transfer RNA will be kicked out and released during translation. First, I'm going to describe what initiation looks like in prokaryotes, and then provide details that distinguishes it from eukaryotes. Yes. What that means is that in the empty site, you're going to have a transfer RNA that has no amino acid on its acceptor stem. So basically, you can think of it as an empty transfer RNA. So maybe a good way to think about it is that the E can stand for two E's. One, it's an empty transfer RNA with no amino acid, and that's where it exits. And so during translation, whichever transfer RNAs have contributed their corresponding amino acid to the growing polypeptide chain, they'll move to the E site. They're empty, so now they're going to exit. And so that's typically what's occurring in the E site. It's really not. When we are talking the compartments later on in the lecture, on the A P site. But the E site is for empty transfer RNAs that will subsequently exit. So during initiation, we have proteins that are associated with that we call initiation factors. And they help the small subunit dock onto the portion of the mRNA that we refer to as the ribosome binding site. That ribosome binding site is a consensus sequence that's recognized by the initiation factors. And these consensus sequences are also called Shine-Delgarno sequences. Once we have the initiation factors that are bound there, what will end up happening is that the initiation factors will bind also to the start codon that encodes for a formulated methionine. So we talked about this in the past, how that is a PAMP, going back to our immunology lecture. Formulated methionine exists only in bacteria. We have a start codon that encodes for methionine. So this is one way in which bacteria and eukaryotes are different. And so once we have the binding of the initiation factors to the transfer RNA, it'll help dock it, and that's going to recruit the subsequent larger subunit. And this is using GTP to help mediate these interactions. And so energy from the GTP is used to help bring in the larger subunit and to complete the assembly of the entire ribosome together. So big picture takeaway here, these are prokaryotes. There are initiation factors that help bind to this ribosomal binding site, the Shine-Delgarno subunits, if you want to use the longer, more interesting name. and they're also going to bind to the transfer RNA to help bring in that first one to create the start, to encode for the start codon for the formulated methionine. And they're also going to help contribute by bringing in the large subunit to complete the assembly of the ribosome to begin and initiate translation. In eukaryotes, just like I mentioned a moment ago, our start codon does not encode for formulated methionine, but rather methionine. Yes, question back. Oh, sorry, Edward, right? Go ahead. Sorry, you're saying, so I think when it says formulated methionine binds to start codon, it's referring to the transfer RNA that corresponds to formulated methionine. That's a good question. That should be, I'll ensure to change the wording to correct that. Miranda, do you have a question? Yeah. Yeah. So again, we have methionine that our start codon encodes for. Prokaryotes have a formal methionine or a formulated. We have slightly different initiation factors that we refer to as EIFs, eukaryotic initiation factors. We also do not have a ribosome binding site. We do not have Scheindel-Garneau sequences, but rather what ends up happening is that there's a specific EIF, eukaryotic initiation factor, that recognizes the 5' cap. And after that, what it'll do is it'll enter into what's referred to as a forward search to go from the 5' cap and look for the start codon. And so what will occur is that there's a complex that forms of the EIF with a GTP, and that it forms a complex with the transfer RNA that encodes for methionine at the start codon. And that'll end up recruiting the small subunit to bind there. And this complex will help begin to initiate at that start codon and then recruit. Again, there's the GTP hydrolysis that helps with the recruitment of the large subunit. And this is what initiation looks like in eukaryotes. So this is are there any questions about initiation? Yes, Marina. Correct. And what will end up happening is that from the 5' cap with a small subunit, they'll begin the search for the start codon. And once that occurs, the transfer RNA will the transfer RNA from methionine will stop there and interact with that codon at that site. And then there will be a subsequent GTP hydrolysis that recruits a large subunit to complete the assembly of the entire ribosome protein or protein RNA complex. And then we're going to into elongation. Are there any other questions about initiation? Yes, Anirudh, sorry, go ahead.
So initiation factors, like they bind to the ribosome. And then after that, like this is just causing
Yeah, so when you say ribosome binding site, that's only for prokaryotes. So as so the initiation factors will bind to both transfer RNA and the small subunit. And they'll typically bind at the, I'll stop there. For eukaryotes, what ends up happening is that our EIFs, eukaryotic initiation factors, they will bind to the transfer, they'll have some that bind to the transfer RNA, and then some that bind to the small subunit to help mediate the forward search for the start codon. And once that happens, the recognition of the subsequent transfer RNA from methionine with the appropriate codon will initiate a conformational change that will help with the GTP hydrolysis and recruitment of the large subunit. So yeah, so one thing that I think I said this from the very first lecture, I didn't keep up with it. I think I was going to say, like, whenever there's a binding event, it's conformational change. So what's always helpful to keep in mind is that whether it's GTP hydrolysis, whether it's initiation factors, or the creation of hydrogen bonds across nucleotides, these binding events will contribute to conformational changes that make it energetically favorable for other things to potentially come in. So the next step is elongation. This is a really large, intimidating picture, but what I'm going to do is just go through the big picture process, and we're going to try and look at 2 snapshots to understand what happens here and to better understand the different compartments of the ribosome, the A site, the P site, and the E site. So either after initiation or during translation, when we have a growing polypeptide, what's going to happen is we have either our first methionine or the growing polypeptide in the P site. Again, P site, you can think of it as the polypeptide site. Try and use the letters to help remember what the functions are, or to help keep this in concert with like what the story is or what the big picture is. So what'll end up happening is we'll have our transfer RNA that is shuttled by an elongation factor, EF. These elongation factors will carry with them GTP. Once the right transfer RNA is brought over and the right transfer RNA matches up or is complementary to that specific codon, what will end up happening is that the GTPs will be hydrolyzed, the elongation factor will release and leave that matching transfer RNA in the A site, the active site, or the acceptor site, if you will. And so what ends up happening is that the subsequent amino acid at the A site with that transfer RNA, either the growing polypeptide or the methionine after initiation will then be, there'll be a polypeptidyl transferase that mediates adding the growing polypeptide or that first peptide, first amino acid to this amino acid in the A site. And so this goes back to the earlier slide where I talked about the transferases, where there's an ATP that's hydrolyzed, and that energy will be used to create the peptide bond between amino acids. That's where that energy is used at this point, where the amino acid, the growing amino acid in the P site, will be added on to the one single amino acid in the A site. And so what ends up happening now is that there's another GTP. You can kind of envision it as like a gear that shifts the entire ribosome by one reading frame, by one codon forward. So this GTP will be, what will end up happening is it'll hydrolyze this other elongation factor that will shift the entire ribosome over. So what was in the A site and the P site are now in the P site and the E site. This empty transfer RNA will now exit, and what has been added on to the newer transfer RNA is now in the P site with the growing polypeptide. Now, all of that might have felt like a fire hose effect all at once, but we're just going to try and take a snapshot look at 2 major parts of this to help understand what's happening. So either after initiation, where we have a single transfer RNA with a methionine, or with a growing polypeptide, we have a transfer RNA sitting in the P site for polypeptide or for peptide. So what's going to end up happening is that there are going to be elongation factors that are going to be bringing in, that have 2 GTPs with them, bringing in transfer RNAs, assessing their complementarity with the codon that's within the reading frame at the A site. When it's the appropriate or matching or complementary anti-codon of the transfer RNA for the codon in the A site, the two GTPs will be hydrolyzed, and that transfer RNA will be accepted and sitting within the A site. And so it's important to note that there are two GTPs that are hydrolyzed here. Once the right transfer RNA is brought in to this corresponding codon, and it's brought in or shuttled in by elongation factors. What this looks like is that we have our large subunit on top, small subunit on the bottom. We have our elongation factor sitting on the side with our RNA in between. Now, once that GTP is hydrolyzed, because we have the right corresponding or complementary transfer RNA, what's going to end up happening is that the peptidyltransferase will help mediate the formation of a peptide bond between amino acid 1 and amino acid 2. And once that peptide bond is formed, it's what you're going to have it added on to the transfer RNA in the A site. Now, with the combination of a GTP and an EFG, this elongation factor, it's going to bind to the large subunit and the small subunit. And as you can imagine, kind of like I don't know if a wagon wheel is the right way to put it, or like almost like a gear, what's going to end up happening is that it's going to shift everything along the mRNA by one codon. And what that's going to do is that's going to shift everything from the A site and P site now to the P site and E site. So to try and illustrate this, we have our transfer RNA being brought in by elongation factor TU or 2. And so it's going to bring it in. And if it is the right corresponding transfer RNA with it, if it's codon, if it's anti-codon on the transfer RNA matches with the codon in that A site, you'll have the hydrolysis of those two GTP molecules. Elongation factor will leave. And now what's going to end up happening is the amino acid that from what was sitting in the P site, whether it's the single one after initiation or a long peptide chain, is going to be added onto the amino acid in the A site. So it could be either one or it could be 50. So if there are, what's going to end up happening is if you have 50 amino acids here, this growing peptide sequence, it'll be added on to this one single amino acid, creating a now 51 amino acid long sequence. So now, after that's been passed over, you can think of it as a bit of a relay. Each transfer RNA is kind of helping with contribution of the ultimate goal of creating the corresponding peptide chain. And so now the elongation factor is going to come in with the hydrolysis of GTP. It's going to create a shift down the mRNA transcript. And now what was in the A site is in the P site, and what was in the P site is in the E site. And as I described before, the E site is where an empty transfer RNA is and where it will subsequently exit. Are there any questions about this so far? Yes.
And then the EF is like it's like a pushes all of that straight in, like
All of that is right, except for one thing. The GTPs that are hydrolyzed do not contribute to the peptide bond formation, but rather it's the energetics from the ester bond between the amino acid and the transfer RNA. That helps with the formation. The energetics from that help with the formation of a peptide bond between the amino acids on the growing amino acid polypeptide chain. No, so the GTP, once they help with the docking of the transfer RNA to within the ribosome and to the. And so then in addition to that, they'll release the elongation factor will then release, let go and leave that transfer RNA with its amino acid there. That was a great question. That's something I should have articulated a little bit more clearly. Rohi, do you have a question? Are you good? Go ahead.
In this diagram is the ribosome moving this way.
It's moving from the 5' to 3' direction.
On that diagram.
Yeah, to the right. So for your sake in studying, this is a slide that kind of details out the entire process for you to refer back to. For your sake and my sake, I'm not going to read out every word on the slide, but this is to help you kind of have something to refer back to when trying to understand the details. The final step in this process is termination. Termination is when the stop codon arrives at the A site. And so, as you can imagine, or not as you can imagine, as we have described up to this point, there are specific transfer RNAs with an anti-codon that is complementary and recognizes a specific codon. For the stop codon, there's a transfer RNA that is associated with that, but rather, a different molecule called a release factor. It is not a charged transfer RNA. What release factor does is it binds at the stop codon. Release factor binds at the stop codon within the ribosome, and instead of transferring an amino acid, it'll actually transfer a water molecule. And what that water molecule does is it helps to create the C terminus or the carboxyl terminus on the peptide chain. And once that happens, once the peptide chain is released, the ribosome or the ribosomal assembly will fall apart and translation is over. So the release factor is what recognizes the stop codon. It contributes a water molecule to create the C terminus on the peptide chain and helps promote the disassembly of the ribosome to terminate translation. Are there any questions there? All right. So yes, go ahead.
Wait, so is the chain growing on that A site, like during elongation?
That's a great question. So yes, it grows on the A site, but is moved to the P site. And so
Oh, so
during elongation. So like each time when the growing peptide chain, it sits in the P site, it is passed over to the new transfer RNA with its single amino acid. But then what happens is then there's the entire ribosome will shift now after elongation factor G, the GTP with elongation factor G, then it'll shift the entire thing down. And now that peptide on a transfer RNA is sitting in the P site now in the middle. This is a representation of what release factor looks like. It slightly resembles the morphology of transfer RNA, but again, is very different. It doesn't quite do the same thing. So some odds and ends, or some last pieces of information for translation. So as the peptide chain is being synthesized, we talked about how folding is determined by hydrophobic interactions, and as the peptide chain is being synthesized, those interactions are present, and so folding is occurring as the peptide chain is being synthesized. To keep an energetic inventory of what's happening during translation, it requires one ATP and three GTPs for each amino acid that's contributed to the growing polypeptide. What's also important to note is that as one ribosome is, this is not drawn to scale. This is not how much bigger transfer RNA is to mRNA, but typically there are many ribosomes that can be present along a single peptide chain or mRNA transcript. And what this electron micrograph shows is that you can appreciate it based on the length of the peptide sequences here. But from the 5' end at the top right, moving down the transcript to the 3' end, there are many ribosomes that are transcribing along it and creating these little squiggles that are each peptide chains that are being synthesized. And so when there are many ribosomes along a single mRNA, we call that a polyribosome or the polysome. And that is a way that can help maximize the efficiency and expression of that single transcript. When talking about the operon model for prokaryotes, we talked about how they're polycistronic. They have multiple genes within a single transcription unit. What will end up happening is that there are multiple ribosome binding sequences, one that is recognized for each protein. And so when we talk about the lac operon, there's going to be a ribosome binding sequence in front of the sequence that encodes for the galactose permease or the beta galactosidase. So for each of those genes that corresponds to either the enzyme or the protein shuttler, there's a ribosome binding sequence at the beginning of each gene that helps permit the translation of those transcripts. And so that allows for simultaneous translation of those specific units along that transcript in a operon model for only prokaryotes. And finally, for some cellular mRNAs, but mostly for viral RNAs, there are these sites called internal ribosome entry sites. As the name implies, they're sites where ribosomes can come in and enter in on the RNA. That is a way to enable translation in a 5' cap independent manner. There are these consensus sequences that encode for the initiation of translation at these sites that don't require the 5' cap being recognized in a forward search occurring subsequently. So we've talked about in the past how there are instances where DNA mutations or variations can occur. And what this slide is trying to indicate is what the downstream consequences are and the categorizations of the mutations that occur that we currently have. And so in one example, we have the DNA encodes for CTT, the subsequent mRNA encodes for a codon that's GAA, and that amino acid at that for that protein for that codon is glutamine or glutamate, sorry. And so what's important to know is that if you change that middle thymine to an adenine, the subsequent mRNA nucleotide will be a U or uracil, and you'll go from a glutamate to now a valine. And so when you have a change in the amino acid as a result of some mutation, we call that a missense mutation. When it leads to a totally new amino acid, we call that missense. In the middle example, when the mutation leads to a new codon that encodes for the stop codon, we call that nonsense. Nonsense is when, again, there's a stop codon that is a result of a mutation. And as we talked before about the wobble that exists within transfer RNA and how the third nucleotide within a codon can change but still be encoding for the same amino acid. When that happens, when we have a mutation, but it still encodes for the same amino acid, we call that a silent mutation. When again, the end product is still the same. We still get the same amino acid. We call that a silent mutation. When we talked about non-homologous end joining, where there are nucleotides that are kind of added in to help repair that double-stranded break, there can be mutations that are either insertions or deletions. And this actually goes back to Crohn's disease when we talked about the manifestation of that disease that seems to be implicated with an insertion allele or insertion mutation on a specific allele. And so for NOD2, this leads to what we call a frame shift mutation. There is one base pair, excuse me, drink some water. There's one base pair that's either inserted or deleted. And by nature of the reading frame being for one codon, that's 3 nucleotides, that one insertion in this example, where we go from TACTT, where we have now guanine that's inserted to TACGTT, this changes the subsequent codons as a result of that insertion mutation. And now all of the subsequent codons, amino acids from those codons are changed. And usually, not always, but usually when we have a frame shift mutation like this, where it kind of like shifts everything, it'll typically lead to a early stop codon. And it'll usually lead to the truncation of a protein, like as was the case with NOD2 from our early innate immunology lecture. So I have this self-test for you all to kind of use to help spark some conversation for you all when I know all of you are meeting together in small groups and huddles and talking about cell and molecular bio all the time. So this is some fodder for you all. So in the final 6 minutes, I will, in a non-rushed fashion like last time, finish up the lecture and talk about how this relates all back to ricin. So ricin is a heterodimeric protein. Again, dimeric means it has 2 subunits, and hetero means there are 2 different subunits. And so we have ricin A and ricin B. Ricin A in yellow is the cytotoxin. But what's important to note is that the cytotoxin is not able to efficiently enter into cells. It relies on ricin B to do that. Ricin B is not the cytotoxin, but rather helps mediate cell entry. And it does that by being a lectin. Lectin, again, you all might remember, you might be thinking back to mannose binding lectin. Lectin is a class of proteins that are sugar binding proteins, for lack of a better phrase. And on our plasma membrane, we have glycoproteins. Those are large proteins with small sugar moieties or monosaccharide moieties on a protein. So lectins typically bind to different sugars. And in this case, ricin B helps form these complexes of ricin on the surface of our plasma membrane because of the glycoproteins on our plasma membrane. And the nature of ricin B being an agglutinin, that's largely something that helps form lots of coagulations or clumpings together. And so in this case, ricin B helps mediate the clumping or combination of ricin molecules at the plasma membrane to help mediate cell entry, because it's also mediating the grouping together and pinching off of the plasma membrane to enter in. And ricin A and ricin B are held together by a disulfide bond. And so that we're going to talk about that disulfide bond and how it contributes to activity once it's in the cell. We haven't talked about protein trafficking yet, but I'm just going to walk through what this looks like. So at step 1, we have the ricin A-B heterodimer binding to the glycoproteins and glycolipids on our cell membrane, and the, for lack of a better phrase, the clumping together, the agglutinin nature of ricin B heterodimers contributes to the internalization or the endocytosis of ricin. Now, once it's been endocytosed, it's within a vesicle now. And so this is still an oxidizing environment. So the disulfide bond is still intact. It's still a heterodimer. They're shuttled, these vesicles now are shuttled in through endosomes. And in some cases now, some ricin is shuttled back to the surface. In other cases, some are degraded in the lysosome. For some lucky few, they're going to continue kind of going backwards through the protein trafficking cascade and go from the endosome now to the Golgi apparatus. We haven't quite talked about the Golgi apparatus yet. It's been referenced in a few pre-lectures, but we're going to talk about it more during our protein trafficking lecture. But what ends up happening is that once it makes it to the Golgi, some will get shuttled back in the normal cascade or pathway of things being shuttled from the Golgi out. There will be some that escape the Golgi into the cytoplasm, where it is no longer an oxidizing environment, but rather a reducing environment. So this will now break that disulfide bond, and now you will have free ricin A in your cytoplasm, which is now able to carry out its cytotoxic function. Again, as I mentioned, what is it about the cytoplasmic environment that allows them to separate? It's the reducing environment that breaks that disulfide bond and allows them to now be a ricin A and ricin B subunits that are separated. And now ricin A is an enzyme that is going to depurinate specific adenines on ribosomal RNA. What's going to end up happening is that depurination is going to alter the ability for elongation factor to come in and bind to the ribosomal subunits. Excuse me. And so that depurination is going to alter ribosomal RNA, subsequently altering our ribosomes, and it's going to inhibit the binding of these ribosomes, the ribosome subunits to the elongation factors, and it's going to stop translation. If you're stopping the elongation factor, you're stopping elongation, you're stopping translation. So that's how ricin acts and why it's so deadly. And typically, one ricin molecule can depurinate up to 50,000 ribosomal RNA molecules. And we have approximately 10 million ribosomes ribosomal RNA molecules per cell. And so with as few as 200 ricin molecules, it can kill a cell. Applications. So besides being a weapon, what are some ways in which it can be useful? Ricin in a research context has helped us to understand some of the mechanisms of translation. It's also helped us understand how proteins can bind to some nucleic acids like elongation factor binding to ribosomal RNA. In cancer, if you're able to be specifically in a targeted way, you can actually use small ricin molecules that can be conjugated to an antibody to have a targeted therapeutic effect to kill cancer cells. If you are if you have any more questions or more interest in translation, I only have time to add one, but Dr. Byers, someone who focuses on ribosomal RNA. We have a lot of great people in the microbiology department that focus on this as well. With that, thanks for dealing with my scratchy voice, and that concludes today's lecture.